All right, as they already said, my name is Gabriella March. Some of you saw me last night, and this is basically the same talk, only longer and with some numbers. And I don't have the advancer. So anyway, let's jump right in. Thanks to my collaborators, I definitely could not have done this alone, and my sources of funding. So here we go. So I study carbonate cemented conglomerates. This is what they look like. It looks a lot like concrete. It's pretty boring. It's hard to get away from because I always see concrete, so I'm always thinking about my rocks. And what I've hypothesized is that if you can't find organic material or living organisms, which is possible that we may not be able to find, most immediately on Mars, maybe you can use isotopes and the mineralogy, sort of a detailed investigation to prove that, or serve as evidence that life existed. So when we started the study, we thought we'd find negative carbon isotopes. We thought we'd find dramatic variability in the isotopes and in the mineralogy on a fine scale, and then mineralogical heterogeneity. So we'll get into that. All right, so I investigated rocks from two different creeks, Adobe Creek and Del Puerto Creek. This is what they look like. They're pretty low flow in the winter time, which is when I went and collected rocks. It looks like somebody dumped a whole bunch of concrete into the rivers. And I want to point out that there are microbial mats all over the place. So a note for you biologists out there. So this is I found these creeks run through the Del Puerto Ophiolite. Adobe Creek runs this way into Del Puerto Creek, and Del Puerto Creek runs down, this is downstream. And so those are my sampling sites. They're actually pretty close together, but could come from very distinct watersheds. And this is where the Del Puerto Ophiolite is located. So it's in California. It's part of the California Coast Range Ophiolite system, so there's lots of Ophiolites up and down the coast, and it's pretty close to San Francisco, and the closest big city is Livermore, so that's where we stay in hotels. Okay, so it's an Ophiolite, and I use that word, and some of you may not know what that means. So we use Ophiolites as Mars analogs, and I don't think this has been mentioned before, but basically, Mars's crust, which is kind of what I'm investigating, it's an analog for Mars. Mars's crust never got differentiated, so it's mostly mafic and ultramafic rocks, which is the same thing we find on Earth in the ocean crust. So we have mafic and ultramafic rocks in the ocean crust. We have continental crust, which is high in aluminum, silicon, things like that. So if we want to study a Mars analog, we really need that ocean crust, which obviously is underwater, which is an issue. But luckily at subduction zones, when the ocean crust gets subducted under the continental crust, sometimes a chunk of that ocean crust, and even sometimes a bit of the mantle, gets shoved up onto the land, which is what we call an Ophiolite suite. And those mafic and ultramafic rocks are sort of a compositional analog for Mars. So there's a lot of interest in the chemistry of the water that runs through there and as an analog for Mars, so it's pretty exciting. So just to give you some context as to why you should even care, carbonates are definitely on Mars. They've identified those. And there are definitely conglomerates on Mars. Those are my rocks right here. These are Mars rocks. Just saying. They look a lot alike. Okay, so dolomite. The carbonate that cement this conglomerate together, one of the carbonates in there is called dolomite. And for those of you that don't know what that is, it's calcium and magnesium with carbonate. And dolomite's been a problem for geologists because forever we could not get it to precipitate out of solution in the lab. And poor Dr. Land did this for 32 years in perfect conditions, and it never happened. But then we found out later that we add microbes to the mix, and some sort of microbial mediation goes on. It's not like a coral or a shell that a clam is making. It's some sort of indirect influence, and there's a lot of theories as to how that happens. It might just be sort of cellular material in the area. We're not exactly sure what they do, but we say that they mediate the formation of the dolomite, and they are necessary in order for dolomite to form in these types of environments at low temperatures. All right, so I'm going to talk a lot about magnesium content in the data, and this is important because calcite is just pure calcium with carbonate, whereas dolomite is calcium and magnesium with carbonate. And it's a solid solution between these two. it's very rarely perfect one or the other. So you'll see high magnesium calcites, which means you have less than 100% of calcite. And then usually I actually found no perfect dolomite in any of my samples. It was always something that was slightly less than 50%, but something mineralogists would still classify as dolomite. So here you have a layer of calcium, layer of carbonate, and on and on. In the dolomite, you have a layer of calcium, carbonate, then a layer of magnesium, carbonate. So it kind of goes back and forth, which is a nice orderly way, but that never happens in real life. It's just theoretical. So magnesium content in the carbonate cement. So here are my actual rocks. In the triangles, you have Adobe Creek, which I'll use throughout the rest of the presentation. In squares, we have Del Puerto Creek. And you'll see that I have magnesium content from 0% all the way up to 50%, which would be perfect, ideal dolomite. And then 100% calcium all the way down to 50%. So Adobe Creek and Del Puerto Creek are there. This is where dolomite would plot, what we would call dolomite, about 36% magnesium all the way up to 50% magnesium in the crystal structure. And you'll see a lot of those points fall into the dolomite range. But then you also have a lot of high magnesium calcites. And obviously there's a lot more of that in Adobe Creek than we see in Del Puerto Creek, but that could be sampling bias, let's be realistic. And then there's also low magnesium calcite. I only have 1 point represented in that. And it may actually be an aragonite, for those of you mineralogists or geologists out there. So the next thing I want to talk about quickly with dolomite is that not only is there variable magnesium content, there's also the concept of order and disorder. So this again is the ideal dolomite structure, right? So you have layer of calcium, layer of magnesium, and on and on and on. So an ordered dolomite can have variable magnesium contents, but those magnesium elements will stay confined to those orange magnesium layers. So you could find magnesium here, here, and here, but you also might have some calcium incorporated in there. So that's what we call an ordered dolomite. A disordered dolomite is something that you definitely have magnesium in those magnesium layers, but you can also have magnesium in the calcium layers. So it's a lot more mixed up, which is why we call it disordered. So you could see magnesium in any one of those layers. So here's an X-ray diffraction pattern. Don't get too nervous if you don't know what this looks like. I'm going to give you a pretty simple example. So this is an actual pattern from one of my rocks. It's pretty representative of the whole. And I'm going to overlay an ideal dolomite pattern, right? But this is not ideal dolomite. This is actually a dolomite with variable magnesium content that's actually disordered. So the way we know that is you'll notice that this peak right here, which I'm pointing to with an arrow because this is the one we like to use because it's nice and strong, it's very wide here. And that's an indication that we're getting lots of different magnesium contents in a very small amount of material that we are measuring. So there's a lot of heterogeneity. And then also you'll see that this peak is very offset from where the ideal peak is, right? So here's the peak, and then there's the overlay. And that offset is an indication of disorder. So we know that just based on the XRD patterns that there's lots of different types of magnesium contents and also disorder in the structure. And there is TEM data to back this up if you're interested to look at it with me, but I didn't have time to show it today. All right, so just to review where we are so far, so these are streambed conglomerates. That means they're low temperature. So they're formed at Earth's surface temperatures. It's disordered, which we're pretty sure is modern, because if you give it enough time, it will eventually order itself. So we know it's a modern dolomite. And so what we need to investigate next is the microbial involvement. And so how I'm going to do that is carbon and oxygen isotopes. And I'm only going to show carbon today. So here's an Adobe Creek rock and a Del Puerto Creek rock. Finally, we see some rocks. You'll notice that Del Puerto Creek looks pretty distinct. It's got a lot more, it's a lot more classed, heavy. And so here are the different textures of carbonate that you find within these. So you'll see these laminated, these layers on the clasts, which are pretty beautiful. There's vein fillings that you'll see. There's this fine-grained sort of grayish material that you can't see any sort of textures in there. And then surface laminations, which are nice and thick, but I'm not going to talk about the surface laminations today. So here's what laminated looks like for an optical microscope and also in backscatter electron on the SEM. I like to point out that this is pretty narrow, which is why we had to use a lot of kind of in situ techniques in order to investigate this. Just crushing it up is not really good enough to understand it. So anyway, here are the carbonate layers. And there's a lot of clay mixed in there. So again, this isn't in backscatter. Right here would be a clay layer. We can talk about clays for those of you that are interested in clays if you want to talk with me later. Here's the vein filling, looks very similar. And I like to point out this is 500 microns, which is about the length of that. Here's what fine-grained material looks like, and you'll see it's a lot messier. Clay is kind of mixed in and not in any sort of ordered way. And then you get this sort of smooth-looking appearance in the fine-grained material in the backscatter. So why do I bother with carbon isotopes? So there's 3 isotopes of carbon, 12, 13, 14. 12 is the most common. 13 is a stable isotope, and 14 is radioactive. So we're going to ignore 14 for today. Carbon 12 and carbon 13, we measure the ratio between them. Biological processes preferentially use carbon 12. And so if you have more carbon 12, what you'll see in the ratio is you'll see more negative. As it gets more negative means more biology. Lower carbon 12 is more positive. So here's an example of how organisms can drive carbon isotope ratios. And as it drives more negative, you'll see all these different microbes and that can drive the ratios more negative. So I did SIMS analysis, which secondary ion mass spectrometer, which we can do isotope ratios with 10 micron spot sizes, which is super cool. We can talk about that later if you want. Here's where the spots are, just in case you have a hard time seeing them. And here are the results. So triangles are Adobe Creek, squares are Del Puerto Creek. And what I want to point out first of all is -8 to -13. That's a pretty small range. Here I'm going to split it out by rock and by creek to kind of see if we can see anything. So there's the 2 different rocks from Adobe Creek I investigated. That's where dolomite, calcite, and low magnesium calcite plot. You'll see the first thing I noticed here was I thought that dolomite might have lower isotope ratios, because more microbial involvement, but there actually is no correlation there because the high magnesium calcite's just as low. Del Puerto Creek, if you split it out by rock, you see no correlations. Again, there's the more dolomite than you see in Adobe Creek. And then you split it out by textures, so laminated fine-grained vein filling. Nothing pops out of the data. There's all of it together. And so I just spent all my time, I went through all this, and we didn't find anything interesting in the carbon isotope data, but that doesn't mean that this isn't interesting at all. And what we see is this -8 to -13 actually matters. So that's more negative than you're going to find in a completely inorganic material. But it's not as negative as we wanted to see. We thought there would be some sort of interaction with microbes on a small scale, which you get super negative numbers, which we didn't see, which was kind of a bummer. But it still indicates biogenicity. And just to give you a sense of how homogeneous this material is, that pink circles are where my samples plot, whereas the other samples are fields of different carbonates in Ophiolite suites and other systems. Not always dolomite, but it's super, super tight data field, which is interesting unto itself. And so we thought we'd find negative carbon isotopes. We did, but they weren't as negative as we wanted, and we didn't see that dramatic variability, but we did find the mineralogical heterogeneity, which is pretty interesting unto itself. So there is an evidence of life in these rocks, but it's not a slam dunk. So thanks.
So do we have any questions for Gabriella?
Do you have any idea at all how the microbes are interacting with the carbonate and forming a biosignature, or you just know that something is going on?
We know that something is going on. It's hard to say, and a lot of people have different theories right now. It's kind of the debate in the dolomite literature at the moment. So sometimes I think it might just be the sort of extracellular material that goes along with the microbes that might be influencing it. But basically, you want to separate the magnesium from the water, because magnesium and water love each other, and magnesium can't get into the crystal structure. So anything that dissociates that magnesium from the water and allows it to get into the crystal structure is what's happening, essentially.
You said your negative carbon isotope values were not a slam dunk. I was wondering if you could comment on what the minimum negative slam dunk value would be, and then if you could just give a 1-sentence summary of your oxygen isotope result.
Okay, so We were hoping to see -15 and below. That was kind of the number I had in my head. -10 could be happening with nothing to do with the microbes. So rivers naturally have a negative isotope value because you're getting this input from the surrounding watershed, which all these plants and animals and microbes in the soil, and so you're getting that negative carbon isotope input into the water itself. So something that negative doesn't necessarily have anything to do with the microbes in the lake. It could have something to do with the organics in the watershed. So I really wanted to see things -15, -20, which is what we thought we'd find. We thought we were seeing averages in the bulk scale, but it turns out that was actually what is on the fine scale as well. And then with the oxygen isotopes, I just got the data last week, so we're still working on it, but some of the preliminary stuff we saw was there is separation between the different textures, so fine-grained and laminated material plots in completely different areas. So these are probably different generations of growth. Either they grow in different seasons or at different times of the day, or there's different, we don't know exactly what, but there's definitely some clumping of the oxygen isotopes, which I love to see because it's a lot more fun to work with. So.
Thank you, Gabriella.